Assalamualaikum warahmatullahi wabarakatuh. Good morning, and if it's not morning, good afternoon and good evening. We are from grade 8 and our topic is the Copenhagen Polio Epidemic 1952. In the mid-1952, Copenhagen, Denmark experienced a severe poliomyelitis outbreak. Over 2,100 cases were reported with more than 300 patients developing respiratory paralysis. Many had bulbospinal polio, which paralyzed the breathing muscle, and the hospital only had 7 iron lungs or respirator, which was not enough to accommodate the number of the patients. So, a medical doctor revolutionized treatment by introducing positive pressure ventilation via tracheostomy and manual bagging. This replaced the less effective iron lung and laid the foundation of modern intensive care. There was reported around 200 medical students volunteered manually ventilated the patient by squeezing a rubber bag attached to a tracheostomy tube inserted through an incision just below the patient's larynx. This outbreak highlighted the lack of natural immunity and vaccine, leading to genocide inactivated polio vaccine in 1955.
First, let's look into the characteristic of poliovirus. Poliovirus belongs to Picornaviridae family in the enterovirus genus. It is a small spherical virus about 25 to 30 nanometers in size. Its genome is single-stranded positive sense RNA. It is a non-enveloped virus having only an icosahedral capsid and autocode made of 60 protomers, each made of 4 viral proteins. There are 3 serotypes of poliovirus, type 1, type 2, and type 3. Immunity to one serotype does not protect against the others. As of now, only white poliovirus type 1 still circulates, while the other types was declared eradicated. How does poliovirus infect human? The pathophysiology started from the entry of poliovirus mainly via fecal oral route, usually by ingestion of contaminated water or food. In some cases, it can also spread in droplets or aerosol form. Once poliovirus enters the body, it begins the first stage called the elementary phase. The virus initially replicates in 2 locations, the oropharynx and the intestinal mucosa, especially in the areas called pie patches and tonsils. The virus attached to human cells by binding to a receptor called CD155. After binding, poliovirus enters the cells and releases its genome into the cytoplasm. Poliovirus RNA act like mRNA, so whose ribosome can directly translate viral proteins. One key protein RNA-dependent RNA polymerase replicates the viral RNA genome independently of the host DNA machinery. New viruses are assembled and the cell eventually bursts, releasing them to infect others. The incubation period of poliovirus ranges from 2 to 35 days. After replication, some virus particles are excreted in the feces, while others remain in the gastrointestinal tract. After the initial replication, poliovirus spread to lymphatic phase. Here, the virus moves from the intestinal mucosa to nearby local lymph node, particularly the cervical and mesenteric lymph nodes. As the immune system circulates lymph throughout the body, the lymph now carrying the virus eventually drains into the bloodstream, leading to the next phase, the viremic phase. The virus is now present in the blood, allowing it to disseminate to distant extrarenal tissues throughout the body. In a minority of cases, the virus progresses to neural phase. It crosses the blood-brain barrier or travels through the nerve via retrograde axonal transport to reach the central nervous system. The virus infects anterior horn cells damaging the motor neuron, causing paralysis of limbs. In the bulbar form of poliomyelitis, the virus affects the brainstem, which can lead to respiratory failure and may be fatal.
I will give a quick overview on how innate immune response to the poliovirus, our body immediate and a specific response. Our first line of defense is skin and mucous membrane. Skin, the outermost layer is tightly packed, which will provide the strong physical barrier against the germ. It provides early acid excretion that will kill or inhibit virus on contact. For mucous membrane, it is located in inner lining of nose, mouth, throat, and gut. It will secrete mucus that will trap invading pathogen and also antimicrobial peptide that will destroy the viruses. The next part is the role of interferon and macrophage. Interferon is a special signaling protein that acts like a disruptor that will travel to neighbor cell and turn on the hundred of antiviral genes, which then directly block the viral replication or make the neighboring cell enter the antiviral state. It will activate NK cell to attack the infected cell. Next is macrophage, a large white blood cell that patrol tissue looking for invader and engulf them. It will release cytokine as they form the pathogen and throw more immune cells to the side to amplify the virus genetic material. These are like a built-in alarm system. Once triggered, they will activate the variant and kickstart the whole immune response. Finally, inflammation, which is our emergency response. Infected cell and macrophage will release cytokine and complement that will increase the blood flow and bring in the reinforcement like white blood cell. It causes redness, swelling, and more importantly, heart trap include the virus.
Moving to the adaptive immune response that also played a crucial role in fighting the poliovirus. First, after the virus enters the body, B cells are activated. They initially produce IgM antibodies, which are the first responders that help neutralize the virus in the blood. With help from CD4 helper T cells, these B cells then undergo class switching to produce IgG antibodies, which are more specific and long-lasting. IgG helps neutralize the virus, block its entry into the host cells, and enhance its removal by immune cells. Next, the T cells come into action. CD4 helper T cells recognize viral antigens and release cytokines that coordinate the immune response. Meanwhile, CD8 cytotoxic T cells identify and destroy infected host cells. This is especially important because poliovirus replicates inside the cells, including motor neurons. So the immune system needs to eliminate these infected cells to stop the spread. After the infection is cleared, the body forms immunological memory. Memory B and T cells remain in the system, ready to respond much faster if the virus returns. This is the same principle that underlies polio vaccines, which prepare the immune system without causing disease. However, in rare cases, about 1 in 200 infections, the virus reaches the central nervous system, particularly the motor neurons in the spinal cord. Even though the immune system is activated, it may be too slow or overwhelmed, allowing the virus to cause neuronal damage and paralysis. This could be due to a high viral load, delayed immune response, or individual differences in immune defense.
Vaccine development. There are two types of polio vaccine. First, we have IPV developed by Genosak and OPV by Albert Sabin. The key difference between these two vaccines is that IPV are killed virus, which means the virus is inactive, no replication whatsoever, just being there for our body to fight and build immunity towards it. Meanwhile, OPV use the same mean as smallpox, where you weaken the virus and give it to the patient. IPV is injected either subcutaneously or intramuscularly, while OPV is given orally, and the virus will course through your digestive tract and induce mucosal immunity as explained by the previous presenter. Of these two vaccines, IPV is a safer option for immunosuppressed or immunocompromised people. So then, more details on these vaccines. Let's start with OPV first. Its cost effectiveness and easy to administer, no need for sterile needles and just drops, made it a preferred choice for low and middle income countries. For several weeks after vaccination, the virus replicates in the intestine, it's excreted, and can be spread to others in close contact. This means that immunization with OPV can result in passive immunization of people who have not been vaccinated. This we call herd immunity. However, there is the risk of developing vaccine-derived poliovirus, VDPV for short, because nonetheless it is a live virus. They can replicate and infect cells. The chance of this happening is 1 out of 2.8 million times if PV is given. Continue to IPV, it does not live, so it doesn't replicate or infect cells, reducing the risk of VDPV or vaccine-associated paralytic poliomyelitis, VAPP. 2 doses of this guy are 90% effective against paralytic polio, according to Center for Disease Control 2024. Thus, there is the global transition from OPV to IPV driven by GP. Countries with strong healthcare systems has transitioned to use only IPV, like Malaysia in 2010. Then the thing is, why the countries don't just use IPV in the first place if it is safer? Well, there was a manufacturing mishap when IPV was first mass-produced in America, which was totally the company's fault for not following the strict guidelines and procedure. For more detail on this story, you can scan this QR here. Alright then, with that, I respectfully pass to our last presenter.
Next, we're going to see about the immunology roles, the lesson from Copenhagen polio epidemic. As we know, polio is a serious disease that can cause disability. Vaccines have greatly reduced cases worldwide as WHO recommends all children to get the vaccine. During polio outbreaks, quick teamwork is needed to pick the right vaccines and stop the spread together with WHO and government help. Polio vaccinations dropped during COVID-19, especially in poorer areas. Healthcare teams play a vital role in building trust, educating families, and organizing vaccination campaigns to improve coverage. Since the World Health Assembly launched the Global Polio Eradication Initiative in 1988, global polio cases have dropped by over 99%. Polio vaccines have helped prevent an estimated 20 million cases of paralysis in children. In the 6th meeting of the WHO Standing Committee on April 9, 2025, it was reported that polio cases have increased with 99 wild poliovirus cases. There were 74 in Pakistan and 25 in Afghanistan. There are also 303 vaccine-derived cases. The virus was also detected in wastewater. WHO continues to support mass vaccinations and surveillance, maintaining polio as a public health emergency of international concern. Malaysia was first declared polio-free by the World Health Organization in 2000 after its last reported case in 1992. However, in 2019, the country faced a setback with its first polio case in 27 years, which, according to the Malaysian Pediatrics Association, is already considered an outbreak. Malaysia was then officially declared polio-free again on 11 September 2021. In conclusion, the 1952 Copenhagen epidemic marked a turning point in medical history as it led to the life-saving manual ventilation efforts that inspired the development of the modern ICU. It also reminded the world of the devastating impact of polio and the incredible power of vaccines to prevent it. Today, continuing immunizations, strong surveillance, and global cooperations remain essential in our missions to achieve a polio-free world. With that, we conclude our presentations on the 1952 Copenhagen polio epidemic. Thank you for your attention and time.